Think of your immune system like a highly trained army, right? It's always on high alert, ready to just absolutely blast any invaders the second they show up. But here's the million-dollar question. How does that army know exactly when to stop? In this explainer, we're diving into a literal game changer, a groundbreaking discovery about our immune system's built-in brakes, and the exact microscopic mechanism that keeps us from, well, accidentally destroying ourselves.
Because think about it, what happens when your immune system actually forgets how to stop? An unchecked immune response is, well, it's terrifying. Picture a sports car barreling down the highway with literally no brakes. Without a definitive way to halt the attack, the body just spirals into this deadly hyperinflammation, severely damaging its own tissues. So understanding exactly how our cells pump the brakes, it's not just some academic exercise. It is quite literally a matter of life and death.
Let's get right into it. Section 1, the cellular bodyguards. To really wrap our heads around this braking system, we first need to look at the microscopic bouncers keeping order inside our cells. We call these heat shock proteins, or HSPs for short. You can think of them as a family of cellular bodyguards. Their normal 9-to-5 job is to carefully fold brand new proteins into their correct functional shapes. And they protect those proteins from getting damaged when the cell is under intense stress. You know, things like a high fever, a nasty infection, or even a sudden drop in oxygen. They are the ultimate guardians of cellular order. But as it turns out, not all bodyguards operate the exact same way. I want you to meet HSPA2. Unlike other heat shock proteins that only rush in when the fire alarms go off, HSPA2 is what we call a non-inducible chaperone. Basically, that means it's always on duty. It's constantly hanging around at baseline levels, especially in our skin, brain, and testes. And here's the wild part. Researchers recently discovered that this constantly present protein plays a massive, continuous role in regulating our immunity. Which brings us to Section 2, the immune system's brake. So, a major recent study by Professor Jan Dopping's team, published in the journal Cell Reports, actually mapped out the exact 4-step molecular mechanism of how HSPA2 controls our antiviral defenses. Check this out. Step 1. A virus sneaks into the cell, triggering the defensive alarms. Step 2. HSPA2 steps right in and binds directly to a super important signaling hub protein called TBK1. Step 3. By grabbing onto TBK1, HSPA2 physically blocks a totally different protein called HERK5 from doing its job of activating the system. And finally, Step 4. Because HERK5 is blocked, that alert signal just kind of fizzles out, and the whole immune response grinds to a halt. The real crux of this is that HSPA2 is acting like a microscopic molecular bouncer. It physically stands in the way. It competes for the exact same spot on that TBK1 engine, specifically a spot called the K608 binding site. By just standing there, it prevents the HERK5 ligase from attaching these little activation tags called ubiquitin molecules. And, without those ubiquitin tags, that TBK1 engine simply can't turn on. The production of protective type I interferons just completely shuts down. Now, you might be wondering, why on earth would our bodies want to shut down our own antiviral defenses? Well, it's a super delicate balance. On one hand, this brake is absolutely life-saving. It keeps the immune system from overreacting and obliterating our own tissue with runaway inflammation. But there is a dark side to this. If this brake gets applied too heavily, it suppresses the immune system way too much. It basically hands invading viruses a VIP free pass to multiply and spread unhindered. Let's look at exactly how that happens in Section 3, how viruses hijack HSPA2. Because this built-in braking mechanism is so powerful, viruses, being the opportunistic little invaders they are, have evolved ways to completely exploit it. Heavy hitters like the severe fever with thrombocytopenia syndrome virus, herpes simplex virus 1, and hepatitis B, they've all developed these really insidious tricks to physically isolate or block that TBK1 signaling hub. By interfering right at this exact pathway, they stop our cells from producing those critical type 1 interferons. They're literally cutting the phone lines before the cell can even call for backup. And the sheer scale of this hijacking, it is staggering. To put it into perspective, researchers found that during certain foul adenovirus co-infections, actively ramping up the host's HSPA2 protein caused a massive 21-fold increase in viral replication. Let that sink in for a second. A 21-fold explosion. The virus just multiplies exponentially, purely because it figured out how to slam its foot down on our own immune system's brakes.
Moving on to Section 4, the cost of broken brakes. But hey, this story isn't just about viruses. What happens when this regulatory axis completely breaks down on its own? The systemic impacts are actually devastating, which proves that this protein's influence goes way, way beyond just fighting off a temporary cold. For instance, a complete lack of HSPA2 in the skin leads to this severe skin inflammation, where structural cells mistakenly pump out massive amounts of damaging inflammatory signals. Meanwhile, up in the brain, excessive HSPA2 expression is actually found hanging out right alongside the dangerous amyloid beta plaques that we associate with Alzheimer's disease. So the real-world stakes for this tiny microscopic break are absolutely massive. We have actual clinical data from the UK Biobank showing that individuals with the highest levels of circulating HSPA2 have a significantly increased risk of incident heart failure and cardiovascular mortality. Think about that. A long-term imbalance in this one tiny molecular break can literally strain and eventually fail the human heart. Which brings us to an incredibly urgent question. Can we control this molecular break ourselves? I mean, if an overactive break lets viruses run wild and strains the heart, and an underactive break causes runaway skin inflammation, can medical science step in? Can we artificially press or release this break to treat these devastating diseases? Let's explore that exciting frontier in Section 5, Future Therapies and Drugs. So, researchers are already working hard to translate this discovery of the HSPA2 TBK1 HRC5 axis into precision-targeted therapies. Right now, we're looking at small molecule isoform-specific inhibitors. Basically, these are drugs designed to surgically block only the HSPA2 break without harming the other helpful chaperone proteins. There are also super exciting combination therapies in the works, like pairing STING agonists with HSPA2 modulators. This could essentially supercharge cancer immunotherapies by releasing the break precisely inside a tumor and letting the immune system just absolutely go to town on it. And then there's this one particularly futuristic method involving PROTACs, or proteolysis targeting chimeras. This tech is like a guided homing missile. It flags pathologically elevated HSPA2 proteins and literally tricks the cell's own natural garbage disposal system into selectively destroying them. This could be an absolute game changer for conditions like heart failure, where HSPA2 levels get dangerously high and just desperately need to be cleared out of the body. So, at the end of the day, the discovery of this HSPA2 mechanism is so much more than just a new textbook fact. It is a doorway to an entirely new era of medicine, an era where we actually get to dictate the speed limit of our own immune system. So I'll leave you with this to think about. Will mastering our cellular brakes finally allow us to outsmart both chronic inflammation and the very viruses that try to hijack us?